Vanakkam. Welcome to this course on classics in neuroscience. We have been looking at neural circuits, specifically on the cortical columns and cortical processing. In the last few videos, we looked at the intracellular brain recordings. In this video, we will look at two motor systems distinct from each other, the pyramidal system and the extrapyramidal system, that are responsible for two types of functions, for two different functions. And we will also look at the work of Wilder Penfield, who studied the human cortex and identified the presence of a motor map or a little man in the brain, a homunculus of sorts. In the 1950s, there was this distinction that was made between the pyramidal system and the extrapyramidal system. What is this system? Pyramidal system refers to the cortical pyramidal neurons that send their long axons through the pyramidal tract to make direct synapses with alpha motor neurons in the spinal cord that project to the distal upper extremity for control of fine movements of the digits or the fingers, fine movements of the digits in primates, especially in higher primates, especially in humans. So, these neurons that project directly to the alpha motor neurons controlling the distal upper extremity, which is essentially the hand and fingers. These are called as pyramidal neurons. Another name for this is corticomotor neurons or the upper motor neurons. Those who are interested in this topic can see a video from a different course that I have taught, Neuroscience of Human Movement, in which I have discussed these pyramidal and extrapyramidal system in good detail and the difference between the upper motor neuronal lesions and lower motor neuronal lesions in good detail. Whereas the extrapyramidal system, in this case the cortical connections are made to the brainstem centers that then connect to interneurons in the spinal cord that control the motor neurons, the alpha motor neurons. This is critical because it is useful to make that distinction or distinguish between the motor defects that happen due to lesions at the level of the spinal cord, due to lesions that happen at the level of the midbrain, due to lesions that happen in the white matter, and due to lesions that happen in the cerebral cortex, because these are different types of disorders and the effects, symptoms are different. That means that the therapeutic approach is different. So, it is useful to distinguish between the two. Wilder Penfield studied under both Santiago Ramon y Cajal and Sherrington, and he founded the Montreal Neurological Institute. He was a neurosurgeon responsible for development of surgical or operative procedures for removal of cortical tissue for relief of epilepsy. So, identifying which are the regions that are causing epilepsy or epileptogenistic regions and removing those tissues. During these surgeries, he performed a systematic mapping of the human motor cortex. He confirmed the presence of the motor strip, the so called primary motor cortex, just anterior to the Rolandic sulcus or the central sulcus. Just anterior to the central sulcus was the primary motor cortex, the motor strip. He confirmed this presence, and he also found that stimulating different regions within this motor strip activated muscles in different regions of the human body. This mapping showed that there was a little man present within the brain, specifically within the motor cortex. This little man came to be called as the homunculus the little man, was present within the brain of people, activating different regions within this motor strip or motor cortex, activated different regions in the body. Of course, what he found was that there was a disproportionate representation of the limbs and the face. There was a huge amount of representation of the upper limb when compared with the trunk, for example. This motor homunculus was comparable and parallel to the somatosensory homunculus found in the somatosensory cortex, that is just posterior to the Rolandic cells or the central cells. And so, he also found not just movements were elicited during shocks or stimulation of the cortex, but also memory, that he could elicit memories. This emphasized the critical contribution of hippocampus in memory. This line of work by Penfield attracted brilliant students to the Montreal Neurological Institute. Some of them were Herbert Jasper and Brenda Milner. Herbert Jasper, as you know, went on to lead neurophysiology and electrical measurements, electrophysiology, in collaboration with other students and other workers. Brenda Milner was responsible for bringing to light the condition of patient HM and describing this condition in good detail throughout the next few decades. With this, we come to the end of this video. In this video, we looked at two motor systems, specifically the pyramidal and the extrapyramidal systems, and we also looked at human cortex and the homunculus, the motor homunculus and the somatosensory homunculus, the little man within the motor cortex and the little man within the somatosensory cortex, and how Wilder Penfield performed these studies in motor cortex and identified the homunculus within them. Also, how Wilder Penfield inspired other neuroscientists like Herbert Jasper and Brenda Milner. Thank you for your attention.